This video is on gene expression, and this is just going to take a slightly different look at regulation of gene expression compared to how we went over it in class and how your book goes over it. So I'm starting out at the top at the central dogma of molecular biology, which is just thinking about how information flows in genetics. So we start out with a genomic DNA, and we're going to zoom in on a single gene here. Now this gene doesn't have a lot of regulatory information. All of its regulatory information is really just here from the promoter. So we can get transcription from the promoter, and that will transcribe the RNA coding sequence plus the introns. The process of transcription, making this DNA code for RNA, is performed by RNA polymerase, which is going to actually link together the RNA nucleotides to make a sequence of RNA. So our RNA is going to look like this, where we're going to have our start codon and our stop codon. We've had the intron spliced out so that all that's between these is the coding region. The start and stop codon are flanked on each side by the untranslated regions, 5' UTR and 3' UTR. We've got a 5' cap and a poly A tail put on in post-processing. This mRNA information can be translated by a ribosome into protein. At the protein level, we have a lot of different kinds of regulation. So some of our feedback regulation we thought about at the protein level. And proteins can undergo all these different kinds of post-translational modification. For example, cleavage, phosphorylation, glycolysis, and ubiquitination. One example that we looked at for cleavage is looking at the pepsinogen getting cleaved to form pepsin. And an example that we looked at for phosphorylation was thinking about the different signal transduction cascades that are regulated by phosphorylation signal. So let's get into regulation of gene expression. Part one is transcriptional regulation. And here I'm just going to be looking at the different ways that a transcription factor can affect the expression of a gene. And so this is transcriptional expression. So this is really just thinking about recruitment of the polymerase to the promoter. So here's our gene again. We have here our promoter region. We have our exons 1 and 2, as well as an intervening region, this one intron. But now this time we also have a cis-regulatory region. The cis-regulatory region is just a piece of DNA that's able to bind to a transcription factor, which is going to be a protein. So here we have a transcription factor that has bound to the DNA. Oh, there's my mouse. That's bound to the DNA here. That's our transcription factor. So again, this is a protein that binds to DNA, the blue parts of the DNA. And this is now inhibiting transcription. So that means that it's blocking RNA polymerase from being recruited to this promoter region. Alternatively, a transcription factor can help recruit RNA polymerase to the promoter region. We talked about this in class in terms of using mediator to help mediate any kind of transcription factor RNA polymerase interactions. So here we have activation, and we're going to get the production of RNA, which will eventually be converted into protein through translation. Here we have a transcription factor adding in methyl groups. So this is just putting little CH3 groups right onto that DNA. And it can potentially activate, but usually it's going to inhibit. So usually methyl groups here are going to prevent some other transcription factors from binding that would normally recruit RNA polymerase. So here we get no transcription. And here we have a transcription factor that modifies chromatin. So this transcription factor here is going to be putting modifications onto the chromatin itself by changing the histone code. And so it can either loosen up the chromatin, which would increase mRNA, or in this case here in this little picture, it is tightening up the chromatin. And so that's going to decrease the ability of RNA polymerase to be able to bind to this promoter. So notice in the last one that I completely skipped splicing. In class, we talked about this in terms of exon shuffling. So we did not talk about the regulation of splicing at all. We just talked about it as a means to produce multiple RNA species, multiple mRNA species off of a single gene. So moving on to translational regulation. Here we have translational regulation. We've got our mRNA now. That's our starting point. So this mRNA is going to have its two untranslated regions, a start codon and a stop codon, flanking a protein coding region in the middle. And of course, we don't want to forget our 5' cap and our poly A tail that got jammed onto this RNA after it was transcribed. So I want to be thinking about these 3' UTR binding proteins. I already mentioned some other post-translational modifications, and so this is going to be helping the cell make the decision whether or not to convert an RNA into a protein at all. And a lot of this does get mediated by this 3' UTR sequence. So 3' UTR binding proteins, there are also 5' UTR binding proteins, but from what I know and what I've read in the literature, the 3' UTR ones are much more prevalent. These 3' UTR binding proteins are going to just bind to the 3' UTR. So again, we've got a protein binding to a nucleic acid. Unlike a transcription factor, these are binding to RNA, not to DNA though. And these are going to help to regulate translation. They can destabilize the mRNA, so target it for degradation, in which case you get less protein from this one particular mRNA. Alternatively, they can stabilize the mRNA, and you can see this mRNA here in this little stable conformer, and this is being stabilized by this little yellow binding protein there, and that means that you can make more protein. It's around for longer, so the ribosome can access this multiple times. The binding proteins can also aid in localizing the mRNA. So in this case, this binding protein is interacting with some cytoskeletal elements that are interacting with the cell membrane. And so this is going to tether it to this one little part of the cell membrane wherever we have these purple proteins around. So this can either give us a localized point source of the protein like we saw with bicoid, or it can just sequester the mRNA. It can basically put it in one cellular compartment so that it can't be floating around everywhere. These binding proteins can also recruit translation factors. So here these translation factors are in purple. And basically once you have these translation factors, you're more likely to get binding of the ribosome. And so that's going to increase protein production. And then, of course, they could inhibit translation factors. So that's what we're seeing here. Here we have our binding protein that's preventing these translation factors from binding. And so that's going to decrease the amounts of times that the ribosome is going to be able to initiate translation. And that's going to lower the overall protein levels. Okay, I hope that this slightly different view of gene expression is helpful to you. Please use the resource that is most useful to you, the book, your lecture notes, this little video, or you can always come to my office hours and we can chat about gene expression there.